This video will demonstrate the steps required to design and order all necessary reagents for creating a single nucleotide polymorphism, or SNP, in a target transcript using the Invitrogen TrueDesign Genome Editing tool. After logging into Thermo Fisher Connect and accessing the TrueDesign Genome Editor, you have a number of experimental options to choose from. For this workflow, we will choose SNP Edit and click Next. On the next screen, you'll see 5 species to choose from. Select the host organism for your SNP experiment and click Next. You'll then select Gene Symbol Entree ID as an input for how to identify your gene of interest. The other available options will use your input to navigate to the nearest transcript in the selected genome. Click Next. When you begin typing in the gene identifier box, a filtered drop-down list will appear. Select your gene of interest and click Search Gene. The next screen will display all of the protein coding transcripts for your selected gene. If there's more than one displayed and you aren't sure which one to select, you can click the Transcript ID hyperlink to be taken to the NCBI website, where you can conveniently visualize the transcript maps. Once you've made a selection, click Edit to proceed to the Sequence Editor view. The Sequence Editor will display the entire transcript's topology along the top of the screen, with a zoomed-in sequence level view below. You can use these views to navigate to the region where you want to edit your SNP. In the Sequence Editor, click on a single nucleotide as you might in a text document. A selection box will appear prompting you to select the replacement nucleotide. Click Mutate. The view will update to display an edit list to indicate the nucleotide change and its position. You can undo the change by clicking the blue Undo arrow. At this point, you also have the option to introduce a second edit within a 30 base pair region of the SNP edit. You can choose a 30 base insertion, deletion, or a second SNP. When your edit list is complete, click Design. This step might take a few minutes while the software searches the region for available PAM sites and assesses the guide RNA designs for efficiency and specificity. When complete, you'll see a table with 2 tabs, CRISPR and TALEN targets. Each row in the CRISPR table displays a CRISPR guide RNA design, its PAM site, its score, which is a weighted score for efficiency and specificity, the number of predicted off-targets, the edit site distance from the CRISPR cut site, and the DNA oligo required as a knock-in donor template. The display will show the DNA donor above the complementary DNA and the resulting amino acid sequence. Green checkboxes indicate recommended guide RNAs due to the score and proximity to the insertion site. Visualize the guides on the Sequence Editor display by clicking anywhere in the row. To view the TALEN targets, click on the TALEN tab of the table, and similar information will be displayed for each TALEN pair. To select one or more guides or TALENs to add to your experiment, use the checkboxes in the table and click Summary. The Summary page will display all of the reagents needed for your SNP editing experiment and give you the opportunity to add additional products to complete your workflow. For a SNP editing experiment, the guide RNA and donor oligo selected in the design step are automatically included. For a knock-in experiment like a SNP edit, the tool will also add Invitrogen TrueCut Cas9 protein and Invitrogen lipofectamine CRISPR-Max transfection reagent. These are added to your experiment by default, but can be deselected from the product summary area, or a different quantity may be selected from the drop-down list. Lastly, you may add additional items such as sequencing primers or positive and negative experimental controls. When you've completed your product selections, you may click Add to Cart for easy one-step ordering of all selected reagents. You may additionally save a detailed report of your experiment by clicking Export Results and Protocol.